So muscle fatigue, if we are exercising or using our muscles at certain time, you will need to stop because your muscles will be fatigued. Why is it fatigued? Depletion of metabolic reserves. You're using your metabolic reserves, which is like glycogen, and you run out of glycogen. So you need to replenish that. Sarcolemma and sarcoplasmic reticulum. This is where we store our calcium. If you're overusing your muscles or abusing your muscles, you can actually damage those structures. Low pH, which is called acidosis or alkalosis. Low pH. Acidosis. So acidosis, why? Because when you do strenuous exercise, you don't have oxygen, so you're not going to burn the glucose until the last molecule or the last atom. So you end up accumulating the product of breaking down of glycogen, which is pyruvic acid. It's going to change to lactic acid and accumulate. And obviously it's called lactic acid, so it's going to lead to acidosis. Beside the muscle exhaustion and pain because of stimulation of the nerves. You stimulate the nerves because of the metabolic products and you feel pain. So you will need a recovery period. And this is the time that you will need all of that to return back to normal. You need to get more oxygen so you can break down the pyruvic acid into until the last energy, point of energy that you can take. You need more oxygen. You need to replenish your reserves. You need to resume the mitochondrial activity. And the most important thing to remember here is oxygen is needed for the mitochondria to work. The mitochondria will not work fully without the presence of oxygen. So that's why if you are like exercising in a treadmill or something or running at certain point of time, you need to slow down and you might need to stop, to completely stop. Why do you need to stop? You need to grab more oxygen. Why? For the mitochondria to work. Why do you have to have mitochondria working? Because this is your only way to break down the pyruvic acid further until the last piece of energy. So one more time, here is glucose entering the cell. Okay? You take this glucose, you break it down into two pieces. Those two pieces are pyruvic acid. And you get just a little bit of ATP so far. Okay, just take it to the cell in the cytoplasm, you break it down, you have two pieces of pyruvic acid. You have a 6-carbon molecule of glucose. It enters to the cell, to the cytoplasm. You break it down. You take 2 ATPs out of it. That's it. And you have 2 pyruvic acids. That's all what you can do so far. Do you want more? The pyruvic acid is still full of energy. Do you want to get the rest of energy out of it? Send it to the mitochondria. And the only way to send it to the mitochondria and break it down further and get the rest of the energy is for the mitochondria to be working. How? You have to have oxygen. So if you are doing strenuous exercise, you don't have the time to catch up your breath, you don't have enough oxygen, so your mitochondria will not be working fully, so you end up breaking down glucose into pyruvic acid. It doesn't go anywhere. The mitochondria is not working. You don't have enough oxygen. More glucose to pyruvic acid. More glucose to pyruvic acid. You accumulate pyruvic acid. It's going to fill the cytoplasm. So how to get rid of it? Let me change it to lactic acid and move it away. Some more. Move it to lactic acid. More glucose. Pyruvic acid. Accumulating. Change it to lactic acid. Move it to the blood. That will accumulate and lead to acidosis, and that's going to stop you at certain point. So what's Cury's cycle? Cury's cycle is just what I was talking about, which is removing and recycling lactic acid by the liver. Convert it back to pyruvic acid and then return it back to glucose. So here is what's happening. This is your cell. Glucose is entering. And glucose is 6 carbon molecule. Okay? So C, 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 C. Okay? Irrespective of the chemical structure. It's just a 6 carbon molecule. And you are in a cytoplasm. Okay? So you break it down into pyruvic acid. 2 molecules of pyruvic acid. That's in the cytoplasm. You don't need oxygen at this time. Are we following? Get the glucose entering the cell in the cytoplasm now. You can break it down into 2 pieces and get 2 ATPs. You don't need oxygen at this time. Not yet. If you want to break pyruvic acid more, send it to the mitochondria. If you do so, you're going to get 34 more ATPs. This needs oxygen. You don't have enough oxygen. What's happening now? Glucose entered to the cytoplasm. Break it down into pyruvic acid. Try to go to the mitochondria. It doesn't go. It doesn't work. The mitochondria doesn't have oxygen. Pyruvic acid accumulate. So how the cell is going to deal with that? The cytoplasm is going to start to fill up with pyruvic acid. How to take care of this situation? Let me move it away, change it to lactic acid. Okay? So that you empty the cell, more glucose enter, break it down to pyruvic acid. The mitochondria will not work, will not take it. Change it to lactic acid. Change it to lactic acid. Change it to lactic acid. Lactic acidosis. So lactic acidosis is a problem. Acidosis, we know that the regular or the normal pH is between 7.35 to 7.45, right? You have to keep it within that range for your body to be working fine. So lactic acid will start to accumulate. The liver will say, okay, I cannot wait. I have to take that lactic acid and change it back to pyruvic acid. Why? To empty the space. Maybe by the time we do so, which is Curie cycle, maybe by the time you change it to lactic acid, go to the liver, change it back to pyruvic acid, and maybe to glucose. By the time I do so, maybe you slow down and you get some oxygen so the mitochondria will start working. So what's Curie cycle? It's this part. Taking the lactic acid to the liver, change it to pyruvic acid to glucose. Now what's the oxygen debt? The oxygen debt is if you are exercising on a treadmill or something, once you stop, do you return back immediately to your regular breathing? Or right when you stop, you still go like, even though you stopped exercising, right? Until you go like, after some time. You are trying to replace the debt. You have oxygen debt. So what's the oxygen debt? The oxygen debt is the debt of oxygen right after strenuous exercise or exertion. So you need to get the debt. Heavy breathing to get the debt, and then you slow down and return back to normal. So this part, did we use oxygen here? Did we use oxygen? Are we following? The first part, are we using oxygen yet? We didn't use oxygen. What do you call it? Anaerobic. An means no. Aerobic means oxygen. Anaerobic means we didn't use oxygen. So this part is called anaerobic. Mitochondria. Can the mitochondria work without oxygen? So you call it aerobic exercise. So if you give yourself the time to breathe, if you're doing light exercise, the glucose will enter the cell, go to the cytoplasm, break down into 2 pyruvic acid, you get 2 ATPs out of it, and then the pyruvic acid itself go to the mitochondria in the presence of oxygen, and you get the rest of energy that you need. So you're fine. You do strenuous exercise. You don't give yourself the time to take the oxygen. So glucose still enter the cell and do the anaerobic part without needing oxygen. You can do it anytime. So pyruvic acid, you split the pyruvic acid into two pieces. That's it. That's the anaerobic part. That's going to accumulate, change to lactic acid to move and empty the space for more pyruvic acid, for more glucose to split into pyruvic acid. So lactic acid, go to Curie cycle, change back to pyruvic acid and glucose. Okay? Do you have oxygen yet? Not yet. Okay, change it back to lactic acid. Go to the liver Curie cycle, return it back to pyruvic acid. Do you have oxygen yet? Yes. Okay, go to the mitochondria, complete the cycle, which is the second part, and this is called the aerobic part. Aerobic part means the part that you have enough oxygen. So this part is anaerobic. You can do it all the time, and this part is called aerobic. The aerobic part is the part inside the mitochondria, which is the TCA and electron transport chain. And this part up here is called glycolysis. Okay? So the anaerobic part is called, what's glycolysis? Glycolysis. Glyco means glucose. What's lysis? Breaking down. So this is just breaking down glucose. 6 molecules, break it down into 2 3s. Glycolysis. If you have oxygen, send the 3 molecules to mitochondria, break it down further, TCA, move the electrons around, electron transport chain. Okay, the energy that you produce when you break down glucose into pyruvic acid, and this energy, most of it will be lost as heat. And that's why if you're exercising, you see your body temperature is going up, right? You feel heat, you feel that you start to sweat, right? Why? Because you lose the vast majority of this, like 70%. You lose it in the form of production of heat. The hormones that work on the muscle metabolism are growth hormone, testosterone, thyroid, and epinephrine. And all of that stimulate. Okay? We don't need to know a lot of details about this. It all stimulate. Stimulate your muscle, stimulate your metabolism. Now, what's the difference between power and endurance? Power is the maximum amount of tension produced. Means I have more power in this muscle than this muscle because I can produce tension more here than here. This is called power. Endurance is the amount of time. So I have more endurance in my lower limb muscles because I can stand up for hours, right? So that's more endurance, irrespective of the power. Do you have both of them? Maybe. But power is one thing, endurance is something else. Endurance is how long can you stay? And this depends on the physical conditioning. If you're exercised, you can stand up for an hour. If I'm more exercised than you, I can stand up for 2 hours and so on. Or you can run for half a mile, okay, and then you have to stop. But I can run for 2 miles. Why? Because this is called physical conditioning. So you start with half a mile and for a week, and then you start to do 3/4 of a mile, and then more and more. This is called physical conditioning. Means you train your muscle to do so. There are three types of muscle fibers: fast, slow, and intermediate. The fast have large diameter and large glycogen stores and some mitochondria, and it contracts quickly, and that's why fast to contract. This is why it's called fast fibers. So fast fibers, the first type of skeletal muscle fibers, they have large diameter, large glycogen reserve, and they contract fast contraction, strong and quick. Slow type, these are the small diameter, the tiny ones. So, and they are called slow fibers because they are slow to contract. And they contain the myoglobin, which is similar to the hemoglobin. Both of them are for storage of oxygen. Hemoglobin is for storing oxygen. Myoglobin is the equivalent in the muscle that's used for storing oxygen. The intermediate is somewhere in between. So this is the slow, this is the fast. So this table can summarize what we talked about, and this is important table. It's summarizing those together. This slow, intermediate, fast. So obviously, slow is slow and small. The fast is fast and large. So obviously, the fast will produce more tension, high tension versus low tension. Intermediate is just in between. So if you remember the slow and fast, you remember the intermediate. Contraction speed, obviously slow versus fast. Fatigue resistance, slow, even though it's slow, but it have high fatigue resistance. So it is slow to contract, but you can use it for some time. The fast is fast to contract, you can use it right away, and it's strong, but you can maintain it for just a little bit of time, less time. The slow ones will have more capillaries and high contents of myoglobin, where the fast has just few. The slow is going to use lipids, carbohydrates, and amino acids, while the fast use carbohydrate only. That's why you can use it for just a little bit of time, because you can use only carbohydrate for it. But for the slow ones, you can burn your fat and your lipids, your fat and your amino acids. The other name for slow is type 1. Intermediate is called type 2A. The fast is called type 2B. Okay, and this is an alternative name that's important. So the slow is called type 1. The intermediate is called type 2A. The fast is called type 2B. Okay, and the fast is the white type of muscles. So if you like when you eat chicken or something, you see some white meat and some dark meat. The white meat is the fast one. The dark meat is the slow one because it contains more myoglobin. So white meat is the fast one, like the chicken breast. The red meat is like the chicken legs, which is the slow ones. And our muscles, this is for animals. Our muscles are mixed. We have mixed both of them, not like animals. The animals have, the breast is white. The legs are red. Our muscles are all pink, meaning it's a mix of those two. Muscle hypertrophy. Hypertrophy. Trophy means enlargement, increase size. Hypertrophy, increase the size and number. So this is when the muscles grow, muscle hypertrophy. And this is like the muscle builders. What the muscle builders have? Hypertrophy. Hypertrophy. Or increased size and number of the muscle fibers. So you start with your muscles like this. You exercise, your muscles get larger and larger and larger, right? This is called hypertrophy. Muscle hypertrophy. And this is come by heavy training like muscle builders. And you're going to increase your mitochondria and your glycogen reserve. This is called muscle hypertrophy. There is another type, but it's not important for us, just for your information. What we're talking about here is just physiology. So you exercise, weight, lifting for 5-6 months, your muscles grow. This is muscle hypertrophy, okay? There is another type that's not natural, not because of exercise, it's because of another problem, but this is not what we're talking about right now. So I mean, the point here is not because you see somebody having like huge muscles, so they are muscle builders. Sometimes people have pseudo hypertrophy, a problem. They have a problem with the muscles. It looks very large and bulky, and you think they are really strong, and you push them, they fall down. They are very weak. This is pathological condition, but we're talking about the physiological one now. Muscle atrophy is exactly the opposite. Atrophy versus hypertrophy. Atrophy, no trophy, no increase in size, or the opposite, which is reducing size. And this is, we know that use it or lose it, right? This is a rule in physiology. Use it or lose it. You don't use your muscles, you're laying down in your bed for long time, so not using your muscles, your body will say, okay, maybe you don't need all these muscles. Let's take what we can out of it and use it somewhere else, okay? Same thing for the bones. Your bone will become weaker if you lay down and you don't do activity, use it or lose it. You're going to start to lose your muscle strength. So physical conditioning, this is what I was just talking about, which is improving the power and endurance. Anaerobic activity, like the 50-meter dash or weightlifting, and this is using more of the fast fibers. And this will need frequent, brief, intense workout. So you play weightlifting or something for 10-15 minutes and stop and do it again several times for a long time. That will lead to hypertrophy. And this is physical conditioning. So today you can do half a mile. Next week you can do 3/4 of a mile. The next week you can do a mile and so on. So this is called physical conditioning. Aerobic, on the other hand, is a prolonged activities. And this is using oxygen, so you're using your mitochondria. Okay? Like you do some aerobic exercises, like light exercise for an hour or something. Okay? We hear about aerobic, right? You do some aerobic exercise versus anaerobic. Aerobic exercise is mild, light exercise, like you're dancing or something. You do that for an hour or something like that, and this is aerobic. Versus anaerobic, you do strenuous exercise for 10-15 minutes. Okay? This is anaerobic. And this will need cardiovascular training and repetitive training. And again, the most important difference is aerobic versus anaerobic. This is a physical conditioning. So again, use it or lose it. If you don't use it, you don't use your muscle, you don't exercise, you don't walk, you don't run, so your body will think that you don't need your muscles. So let's take the proteins out of it, let's take the storage out of it and use it somewhere else. So the muscles will start to shrink and become smaller. And it can be actually replaced by fibers. So this was for the skeletal muscles. Skeletal muscles are muscles attached to the skeleton. And this is our focus for 233. We need to know a little bit about the other muscles, but this is what you're going to focus on in the lab. Now the cardiac muscles. Let's just know a little bit about it. The cardiac muscles are striated and only in the heart. Was skeletal muscles striated or not? What striated means? Stria. What stria means? Layers. Was the skeletal muscles looks like this, parallel layers? There are some transverse, but the main fibers are running like this, parallel to each other, or was branching? Parallel. So they are striated. Striated means put in a stria. Like if you look on a muscle like this, you will see stria after stria after stria. Okay, or looks like this. This is called striated. So both skeletal muscles or cardiac muscles are striated. If you look at the smooth muscles, you will see them scattered like this. No striation. So skeletal muscles will be like this. Cardiac muscles striated, but they do some branching. We will see that. What's the difference then between cardiac muscle and skeletal muscle? Both of them are striated, yes. But the skeletal muscles are large, the cardiac muscles are small. Why? Because if you still remember, if you studied the previous part, you will remember that the muscle fibers, the skeletal muscle fibers, came by fusion. Here is one muscle fiber, another one, another one, another one, another one. and each one have a nucleus, and they all merge together to make a big one, multinucleated. Do you still remember this? It's one of the slides that have something like this. So the versus the cardiac muscle is just one. Okay, so obviously the skeletal muscle is larger, and because it came from fusion of many fibers, they fuse together, so it's multinucleated. Okay, so this is the first difference. Small, cardiac muscle is small, skeletal muscle is large. Cardiac muscle contain one nucleus because it came from one, while the skeletal muscles are multinucleated because it came from several fibers. There is no triad. Do you remember the triad? There is no triad in the cardiac muscles. There is no terminal cisternae. Do you remember that? The blue. This is not in the cardiac muscles. The cardiac muscles are aerobic only. The skeletal was both. It could work both ways. And the most important difference is intercalated discs. Intercalated discs. Intercalated discs are the discs in between the muscle fibers. Goes like this. Like here is one fiber. There are discs in between those. Intercalated discs only in the cardiac muscles. Okay, so what's intercalated discs? These are the contact points between two adjacent muscle fibers. Here is one intercalated disc in between. This is only in the cardiac muscles. Why do we have it? Maintain the structure. Most important thing is conduction of action potential. You got the action potential in this muscle fiber. You want it to transfer to this one. You have the intercalated disc in between. Okay? So what's the most important thing about the intercalated disc? Only in the cardiac muscles, and it transfers the action potential between two muscle fiber. Conduction of action potential. So here is the cardiac muscle. You don't see the blue, the cisternae. They are obviously parallel to each other. So it's striated, small, and so on. I think we have a table to summarize that at the end, so we'll take a look at it. For the cardiac muscles have the automaticity. What's automaticity? Contraction without neural stimulation. Do you remember how the skeletal muscles have a nerve that goes all the way and branch and feed the muscles or innervate the muscles? This is not the case for the cardiac muscles. The cardiac muscles have the SA node within the muscle fibers. This is called automaticity. You don't have to provide them with nerves. And basically, I don't know if you guys did general biology or something before. Did you do like frog heart or rabbit heart or something like that? If you take the heart completely out of the rabbit or completely out of the frog, it will still contract. Did you ever see something like that before? It will still. Why? It doesn't need neural innervation. It does have like a battery inside, which is the SA node. It's inside the heart, and I remember doing that in the very beginning of medical school. We have to take the heart of a rabbit or something, and we like attach it in both ways and see the contraction on a graph. So you take it out, just the heart, nothing else, and you leave it still contract for some time, and the only thing that stops the cardiac muscles is when they run out of reserve, they run out of energy. Okay, so if you provide them the cardiac muscle, the heart somehow with some nutrients or oxygen, something, they will continue to work. So this is automaticity. Skeletal muscles, get a skeletal muscle and put it like on a table or something, it will not work. You have to provide them with nerve. So this is automaticity. Both of them are controlled, meaning if you leave the heart by itself, it will contract. Okay? If you cut all the nerves that go to the heart, what happened to the heart? still contract. So what's the nerve doing? The nerve is going to change the pace. So if you leave the heart by itself, maybe it's like 60. Can you make it 70? No. 50, 40? Nothing. It will just stay like this. But the nerves will come to it. Sympathetic can speed it up, and parasympathetic can slow it down. Leave it all by itself, it will still contract, but you cannot control it. So it is controlled by the nervous system, and the contraction time is like 10 times as the skeletal muscles, so that they contract for a long time. And we know that the heart contracts 24/7, right? It never stops. So the contraction is extended, and there is no wave summation. There is no wave summation. This is important, right? If I'm carrying something that's lightweight, I don't need much wave summation. If it is heavier, I need some summation. If it is more, I need tetany. But does the heart change? Is the heart carrying anything? It does not. It's either speed up or slow down. It can increase the force of contraction a little bit, but no wave summation. We do not do that. Why? Because if the heart go to tetany, the tetany will be like this. This is speeding up. This is the heart. Speed up. More. More. It will go like this. It will not push the blood out. So we don't want that to happen. This is tetany. How to prevent that? There is no wave summation in the heart. Why? Because we have longer refractory period. We talked about the muscles in details. Do you remember the refractory period? The refractory period in the heart is really long, that you cannot, like, this is the refractory period. The signal come here, the muscle will not respond. Here, not responding. Here, not responding. It will respond only at the end, like this. Versus this. So this is the regular muscle. You send the contraction here, or the signal here, it will respond. For the heart, no, 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 until you come here. So the refractory period is longer in the heart. And that's why there is no tetany or wave summation. Smooth muscles. What is the smooth muscles? Anywhere that's not attached to the skeleton or in the heart. You name it. You don't have to even memorize. Ask yourself, is it attached to the skeleton? Yes, skeletal muscles. Is it in the heart? Yes, cardiac muscles. Is it anything else? That would be the smooth muscles. So how about the urinary bladder? Is it cardiac or skeletal or smooth? Smooth. How about the blood vessels? Smooth. Anything is smooth if it is not attached to the skeleton or within the heart. Sphincters. Anything else is smooth. The smooth muscles are not striated. There is no layers. Stria means layer. It's not layered. It's not parallel. It's scattered. So it looks like this. Do you see any striation here? It's completely random. Smooth muscles are long, cylinder, and spindle-shaped. Single nucleus. How many nuclei in the cardiac muscles? One. How many in the skeletal muscles? Many. Many. We don't count, it's many. Okay, you don't have T-tubule or myofibrils or sarcomeres. None of those. There is no tendon or aponeurosis. What's the difference between tendon and aponeurosis? Both of them attach the muscle to the bone. What's the difference? Right. The tendon is a bundle. The aponeurosis is a sheet. That's it. Both of them do the same function. Both of them are connective tissue. Both of them attach the muscle to the bone. What's the difference? It looks different. That's it. A bundle or sheet. So the smooth muscles don't have any of those. The smooth muscles have excitation contraction coupling. So excitation comes from a nerve, and that's coupled by contraction. And the calcium binds to here to calmodulin. And this is different. What the calcium attached to in the skeletal muscles? Do you remember that? When calcium comes out of the sarcoplasmic reticulum, it's going to attach to tropomyosin. Do you remember this? It attach and uncover the active site. Do you still remember that or not? A little bit. You didn't study during the spring break, obviously. Okay, so instead of attaching to tropomyosin and revealing or uncovering the active sites, it actually attached to calmodulin, which is different. You use the ATP. Myosin light chain kinase enzyme is activated here. Length tension relationship. And the control of contractions work as multi-units. Okay. Okay, so this table is important. That's going to summarize all what we need to know. Okay? You can go through the details, but this is summarizing everything. Okay? So if I ask you for the nuclei, skeletal muscles, is it uni or multinucleated? Skeletal. Nuclei, is it one or two or more? So it's multi. How about the cardiac?
One.
Smooth muscles?
One.
One. You have a sarcomere in the skeletal muscle. You have a sarcomere in the cardiac. You don't have sarcomere in the smooth muscles. Sarcoplasmic reticulum, you have cisternae here. You don't have cisternae in the cardiac muscles, not in the smooth muscles. And then in the skeletal muscles, you have neural or neuromuscular junction. The heart muscles, automaticity. The smooth muscles, automaticity. But the hormones will affect them. The source of calcium, sarcoplasmic reticulum. In the skeletal muscles. But in the cardiac muscles, it's a sarcoplasmic reticulum also, plus some extracellular fluid. Same thing for the smooth muscles. Calcium regulation, troponin, troponin. And thin filament, same for the cardiac muscles. Smooth muscles have calmodulin, not troponin, calmodulin. Contractions, rapid onset, slow, and the heart and the smooth muscles. Okay. Which one of those is branching? The heart is branching. Which one is striated?
Skeletal. All of them but the smooth, right?
Skeletal and cardiac. Both of them are striated. Okay? Okay, so this is summarizing the differences between those two, between the three types of muscles, and that will be it for this chapter.